Yeah, everybody, welcome back to this session 3, which is a postgraduate speed session, and it is being chaired by Dr. Zulfia Muhammad Musa, and I hand over to her. Over to you, Dr. Musa. Thank you.
Thank you, Prof. Good day and welcome to the Postgraduate Research Speed Session 1. In this session, our postgraduate students will be sharing their research with us in just 2 minutes, which I'm sure you all agree is a very great feat, so well done to them all. There's no live Q&A slot for this session, and unfortunately, due to the connectivity issues, I'm unsure if all our speakers are live. However, some of them may be, so if you feel the need, please interact with them via the chat function. Without further delay, let's proceed to the presentations. Thank you.
All protocols are duly observed. And by name, Sumana Victory, I will be presenting on the topic evaluation of the effect of probiotic and ascorbic acid on the adverse effect of heat stress in broiler chickens. By way of introduction, global warming has been a major factor in inducing heat stress due to climatic change across the globe. Heat stress resulting from high environmental temperature has been an impediment to the productivity and profitability of poultry farming, hence the need for its mitigation. 56 broiler chickens will serve as the research subjects. They will be divided into 4 groups of 14 each. Group 1 will serve as the control, Group 2 the probiotic administered, Group 3 the ascorbic acid administered, and Group 4 the combination of probiotic and ascorbic acid administered groups respectively. They will be given access to feed and water ad libitum. All administration will be done via feed for 35 days. The effect of these antioxidants in ameliorating the detrimental effect of heat stress will be evaluated via some performance, behavioral, physiological, and biochemical parameters. At the time of this presentation, this research will be almost concluded. It is anticipated that the administration of probiotic and or ascorbic acid to broiler chickens exposed to heat stress mitigates the adverse effects, which will be evident as improved performance, behavioral, physiological, and biochemical parameters. Thank you for listening.
This study was a clinical prospective study that compared antimicrobial efficacy of 4 surgical hand preparation procedures in veterinary students performing averiohysterectomies on dogs. Participants were allocated to 4 groups, of which 3 groups performed a pre-wash with different hand solutions, followed by an alcohol-based hand rub. Pre-wash solutions included a chlorhexidine scrub, F10 scrub, and a pH-neutral soap. The 4th group omitted a pre-wash prior to performing an alcohol-based hand rub. Fingertips were pressed on agar contact plates at different time intervals, and from those samples, colony-forming units were determined. The results were compared with respect to colony-forming unit log 10 reduction and total log colony-forming units. Following the pre-wash, we found a greater log 10 colony-forming unit reduction with chlorhexidine-containing scrub compared to pH-neutral soap. There was no difference in log 10 colony-forming unit reduction from the baseline to 2 hours after surgery between the 3 groups that performed a pre-wash prior to alcohol-based hand rub. 2 hours after surgery commenced, the group that performed a pre-wash had significantly lower total log colony-forming units than the group that did not perform a pre-wash. In conclusion, performing a pre-wash prior to applying an alcohol-based hand rub had better antimicrobial effects before and for the duration of surgery than not performing a pre-wash at all.
Good morning. I'm going to be presenting a short abstract today on the short-term clinical outcomes of 220 dogs treated with a mini hemilaminectomy for thoracolumbar disc disease. Okay, so in these dogs we did 252 spinal decompressions, so some dogs had multiple surgeries performed. We looked at days to ambulation and days to micturition, so DTA, days to ambulation, DTM, days to micturition. And we compared that to the Frankel score of these patients, as well as the recovery rate for each group. We saw a significant or a direct relationship between a decrease in modified Frankel score and increase to both days to ambulation and days to micturition, with a modified Frankel score of 0 showing a significant increase in days to ambulation and micturition. Our recovery rate was also significantly related to both the modified Frankel score. So as the modified Frankel score decreased, the rate of recovery decreased. You can see for groups 5 to 1, the recovery rate was still relatively high, whereas group 0, the recovery rate of 64% was comparatively lower than the rest of the groups. All of this data we collected is comparable to a similar study performed with a similar materials and methods for a hemilaminectomy. We saw a post-operative complication rate of about 13%. Of these complications, 38% were minor complications that did not require further surgery or affect the outcome of the patient, and 62% were considered major complications, which affected the outcome of the patient or required a repeat surgery, and obviously excess cost to the owners. What we documented in the study is that basically the mini hemilaminectomy technique gives comparable results in both outcome and recovery to the previously published hemilaminectomy technique, and our study was the largest study performed on a cohort of dogs, with the nearest study having just under 100 dogs. Thank you very much. Bye.
Good morning. My topic is feeding patterns of Colocoides at Ornosport. Colocoides midges are vectors of viruses, nematodes, and protozoa. One of the most economically important viruses transmitted by Colocoides midges is African horse sickness virus. The aims of this project were to determine the species-specific feeding behavior of Colocoides at Ornosport, to estimate the efficacy of carbon dioxide traps for the collection of Colocoides in this area, and lastly, to determine whether there is any daytime activity of the Colocoides species collected. Using Ornosport light traps and custom-made carbon dioxide traps, we collected Colocoides at Ornosport for the period of a year. Traps were placed in the vicinity of horses, sheep, cows, and dogs. Using stereomicroscopy, collected midges were identified at species level and were then sorted by sex and parity. Individual blood-fed midges were then analyzed by amplifying the mitochondrial cytochrome B gene to determine the origin of the blood meals. Our study showed that the Colocoides midges at Ornosport show a distinct preference for feeding on horses. The blood meal analysis also showed us that blood-fed midges may stray further from the host animal than previously believed. Interestingly, we also determined that Colocoides leucostictus feed on dogs, whereas they were previously believed to be ornithophilic. It is therefore possible that they feed on other mammals. Our results also suggest that carbon dioxide traps are of limited use in the study of the epidemiology of Colocoides imicula at Ornosport. This is unfortunate, as we need an alternative to light traps to determine whether there is daytime activity of African horse sickness virus vectors in South Africa. Thank you.
African swine fever is a lethal disease of domestic pigs that was first described in South Africa in 1926, in the north of the country. A controlled area was declared and regulations implemented to limit the spread of disease from north to south. This controlled area also was linked to the solvatic cycle, which is a circulation of the virus between Ornithodorus tick species and warthogs which did not become ill. Outbreaks elsewhere within the country were as a result of the movement of infection from the north. The controlled area and measures along with it were successful for many years at limiting the spread of disease from north to south, until 2016, when a series of outbreaks south of the controlled area occurred that could not be linked to the movement of infection from the north. Warthogs have also increased in their distribution as a result of translocation, and Ornithorhynchus tick species are known to be present south of the controlled area. We set out to investigate whether African swine fever solvatic circulation occurred south of the controlled area by testing warthog sera for antibodies to the virus and Ornithorhynchus tick species for the virus. Image number 1 represents a highly endemic area, the Kruger National Park, where there was a high prevalence for virus antibody. In contrast, image number 2, Addo Elephant National Park in the Eastern Cape province, no antibody was detected. Image number 3, interestingly, Makala National Park in the Northern Cape province, multiple sera were positive for African swine fever virus antibody. The Northern Cape province has also experienced sporadic outbreaks of the disease in domestic pig populations, in some instances where the source remains undetermined. African swine fever virus was also detected in Ornithorhynchus tick species, and the genotypes determined were consistent with those that were circulating in domestic pig populations outside the controlled area. We conclude that African swine fever solvatic circulation occurs south of the controlled area.
A study was conducted to determine the effect of gallium, which is a natural garlic-based product, and how it can act as a tick repellent when supplementing via the feed to the animals. 16 Bonsmara cattle were used and supplemented with 2 grams of gallium per head per day, where the control only received the feedlot diet. We then placed Ripicephalus appeniculatus, the adults, as well as Ripicephalus microplus in larvae on different parts of the body of the animal, including the ears as well as the back. The ticks were then covered with a cloth to prevent them from moving around too much on the animal. Once the ticks were engorged and detached, we took them to the lab where they allow them to lay eggs, and then we determined the hatchability of the eggs. There was no significant difference on the effect of gallium on the brown ear tick. However, there was a numeric reduction in number of larvae produced per beef animal when you take into consideration the number of engorged females, eggs produced per female, and the hatchability of the eggs. There was a tendency towards a reduction in the number of unengorged females when gallium was supplemented with the microplus ticks, as well as a tendency towards a reduction in the number of larvae produced per beef animal by almost 42%. This is taken into consideration number of engorged ticks, eggs produced per female, as well as hatchability. There was no negative effect on the red blood cell count when supplementing gallium. This is important to determine, as high levels of garlic or onions may cause anemia, and we had to determine there was no negative effect. Thank you.
The first official outbreak of FMD in Malawi was reported in 1957. However, very little is understood concerning the dynamics and drivers of FMD in Malawi. We then decided to carry out a space-time analysis to detect the FMD clusters in Malawi, which was followed by a matched case control study to investigate the risk factors of FMD. Risk maps were also created using ordinary kriging to establish the trend of FMD risk in Malawi over the period of study. In this study, we identified SAT1, SAT2, SAT3, serotype A and O as the main circulating serotypes of FMD in Malawi, with SAT2 being the most prevalent. We then identified 2 main FMD clusters, one in the northern region, the other in the southern region. And these clusters were identified close to international borders and wildlife protected areas, which we could assume were their main source of infection. Higher beef cattle density, higher pig density, and increased distances to wildlife areas increased the likelihood of detecting FMD in Malawi, while increased distances to roads and international borders did decrease the likelihood of detecting FMD in Malawi. In the past, livestock in the northern and southern region were observed to be at a higher risk of infection with FMD as compared to livestock in the central region. However, in 2019, the livestock in central region were observed to be at a higher risk of infection with FMD, which could be attributed to the increase in livestock population in this particular region. We could therefore conclude that all regions of the country are now at risk of FMD infection. These findings therefore provide an insight into the pattern of FMD as well as the risk factors of FMD in Malawi, which should promote informed decisions and control of FMD in Malawi. Thank you.
Hello everyone, you are welcome to my presentation titled Histomorphometry of Seminiferous tubules in 4 reproductive cycles of Japanese coils, Cutinus japonica. I am Dr. Musa Zakaria, a fourth-year PhD student in the Department of Anatomy and Physiology, Faculty of Veterinary Science, University of Pretoria, South Africa. Seminiferous tubules are components of the testes, where germ cells are transformed into mature spermatozoa in a process known as spermatogenesis. In avian species, spermatogenesis has been scantily evaluated, even though they constitute more than 50% of all the vertebrates, and so measurement of seminiferous tubule components are among the most important parameters in estimating efficiency of spermatogenesis. And so the study is aimed at evaluating seminiferous tubule components such as seminiferous tubule diameter, seminiferous tubule epithelial height, and seminiferous tubule lumen diameter in 4 reproductive cycles of Japanese coils. A total of 28 beds were used for the study, comprising of 7 beds per each of the reproductive cycles, namely prepubertal, pubertal, adult, and age beds. Light microscopic evaluation of the cross-section of seminiferous tubules, as you can see on the screen in figure A and B, showing how the components were measured, while figure C is a bar chart showing the mean seminiferous tubule parameters, in which a significant difference was observed in the age group by presenting a lower seminiferous tubule epithelial height and a wider tubular diameter, which could be due to a high rate of germ cell apoptosis and a fall in androgen levels, which are common indicators of aging, as seen in both avian and mammalian species. I am grateful to my supervisor, Professor Lindy Jemaga, for her mentorship, our HOD, Professor Joseph Chamnora, and the members of avian research group. The study was supported by National Research Foundation, South Africa. Thank you for listening.
Hello everyone, my name is Muhammad Ibrahim. I'm presenting to you about our study about those related morphological changes in the epididymal region of sexually active adult male Japanese quail exposed to diambutyl phthalate commencing during the prepubertal stage. This study under the supervision of Dr. Jung Williams and Prof. Kritu Potha. Materials and methods. 25 prepubertal Japanese quail divided randomly into 5 groups. The first group is a control group in which administered corn oil as vehicle only. The other treatment group administered different doses of DPP for 30 days. Light and transmission electron microscope as well as histometrical measurement of epithelial height were done. The results. There were no obvious changes observed in the rete testes. However, several lesions observed in the proximal efferent ducts, such as degenerative changes, cytoplasmic vacuole apoptosis, and autophagy in the quail treated with 200 and 400 mg of DPP. As we can see, this is the control group, and this is the other treatment group. This is the 200. We can see many cytoplasmic vacuoles and degenerative changes in the epithelium of the proximal efferent duct. In addition, epithelial cytoplasmic vacuoles detected in the distal efferent ducts and the epididymal duct in bed treated with 50, 200, and 400 of DPP. In addition, the lumen of the epididymal duct was oligospermic, as we can see compared with the other group, with the control group. The histometrical measurement has shown a decrease in the epithelial height of the rete testes and efferent ducts in all treated group, but not in the epididymal duct epithelium. In conclusion, DPP induced those dependent morphological changes. The proximal efferent duct is extremely sensitive to the effect of DPP. Changes in the epididymal ducts of the Japanese quail treated with DPP could cause infertility. The testicular excurrent duct system of Japanese quail is suitable and sensitive region to monitor the effect of environmental toxins on fertility. Thank you very much for your attention.
Ionophores or polyether antibiotics, often used in the livestock industry for the control of coccidiosis, as well as the promotion of growth and feed efficiency. Unfortunately, due to feed mixing errors and extra label use, ionophore toxicosis sometimes occurs, primarily affecting the cardiac and skeletal muscles, which can lead to the death of the animal. Ionophores are capable of transporting cations across biological membranes, resulting in disruption of the ion homeostasis of the cell. This leads to a cascade of downstream events, culminating in cell death. The MTT viability assay was used to determine the cytotoxicity of the ionophores on these cell lines after 24, 48, and 72 hours exposure. Monensin was the most toxic, with EC50s in the nanomolar range after 48 and 72 hours exposure, followed by salinomycin and finally lasalicid. Microscopic examination of the cells exposed to monensin for 48 hours showed some cells filled with cytoplasmic vesicles. Others became round and detached from the plate and neighboring cells. Immunocytochemistry and immunofluorescent techniques were used to investigate the effects of the ionophores on the cytoskeleton. Figure 3 shows C2-C12 cells stained for desmin after exposure to 1 micromolar monensin for 48 hours. Desmin stained darker around the nuclei of some cells and the intermediate filament network was disrupted. Finally, figure 4 shows actin and tubulin stained green and red respectively, and the nuclei of the cells stained blue. Many of the cells exposed to monensin were round, lacking the long cell-to-cell protrusions seen in the negative control, and with some cells showing slight disruption in the microtubule network. Among others, ionophores causes calcium overload, as well as an increase in the production of reactive oxygen species, resulting in the loss of cell viability, and could indirectly lead to the disruption of the cytoskeleton network. Further research on the method of cell death and the effect on the cytoskeleton is still ongoing.
Bioassay guided fractionation of Senna singuana and its potential for development of poultry phytogenic feed additives. Phytogenic feed additives should have biological activity if they are to be used as alternatives to antibiotic growth promoters. The aim of the study was to evaluate the antibacterial, anti-lipoxidase, antioxidant activities and in vitro safety of Senna singuana fractions and isolated compounds in order to assess their potential for development of poultry phytogenic feed additives. We collected Senna singuana leaves from Chipinge district in Zimbabwe. We then did bulk extraction using 80% methanol. We then moved on to do solvent partitioning with hexane, dichloromethane, ethylacetate, butanol, and water on the crude extract. We then did column chromatography on the ethyl acetate fraction based on its promising biological activity. We obtained 11 main fractions from the first column, and we then ran another column on the A4 fraction, and we obtained 11 subfraction, and we then did preparative TLC on the DD2 fraction. We then obtained a yellow compound, luteolin. We then did in vitro assays on the fractions from the first column and also on the isolated compound. Antibacterial and antioxidant biotography showed that fractions from the first column had active compounds. Quantitative antibacterial activity results showed that some of the fractions had significant antibacterial activity with MIC values of less than 100 micrograms per ml. Regarding anti-lipoxidase activity, all the evaluated fractions were potent and were more active than toxic, indicating anti-inflammatory potential. Quantitative antioxidant activity results showed that the IC50 values of the fractions were comparable to those of the positary controls in both the DPPH and ABTS assays. Based on the notable antibacterial, strong antioxidant and potent anti-inflammatory activities as well as low cytotoxicity of its fractions, the study proposed Senna singuana as a promising candidate for the development of poultry phytogenic feed additives. Thank you.
Thank you to all our speakers. I now hand over to Prof. Penzon, who will conclude the session. Thank you.
Thank you, Dr. Musa. Thank you to our speakers. We head out now to the 30-minute break, and then we'll see you back at the next speed session. Thanks, everyone.